yesterday, I started telling you about this peculiar phenomena. And if you remember, this was one of my slides from last class, where we were discussing things that could be ion-ion correlation. So remember that ion correlation is when we have this electrostatic coupling parameter that's greater than 1, and it often happens if you have a high valency contra-ion, right? But we also discuss things that could happen in this condition where we are not supposed to have this attraction because the electrostatic coupling parameter is a number closer or equal to 0. This happens, for instance, when you have your protein at the isoelectric point, which means that the net charge of your protein is 0. So when I was telling you like that, I showed you some experimental data. So this is one example from what I already showed you. And then I discussed some hypotheses that were made, I mean, how to try to explain it. And finally, I showed you this slide where I said, well, this is a good point to break, and let's continue on Saturday, so tomorrow. That's what I said yesterday. And the paper actually is from 52. You can see that's quite old. But somehow, we have a tendency to don't like to go to the library. So although this paper has been published for quite some time ago, when people were looking to this phenomenon, I mean, they were looking to the experimental data, they somehow didn't check in the library. So that's the first take-home message from today. Spend some time going to the library. It could be a digital library, but go to the library and try to check what others has done before. Because maybe you're just trying to reinvent the wheel, and maybe your wheel is not going to be the proper one, but it could be a square. So be careful. Anyway, back to, again, my hero in science, Kirkwood. You can see that in this paper, he already make a comment that when one or both a pair of protein molecules are at their ionic points, that's similar to the isoelectric point, structure-sensitive electrostatic forces come into play. So he already introduced that some kind of weird phenomena could happen, and if you continue the paper, he can see that he could not test the theory that the statistical mechanic theory that he wrote in this paper because the lack of knowledge of the protein conformation. So he didn't have the PDB and he didn't have AlphaFold. Lucky we have. So we could of course test if what he has proposed in terms of statistical mechanic analysis would make it true, could be true. And just to make, this paper short, of course, there are lots of details. And I guess, if I'm not wrong, in one of the previous schools, I have gone through all the math behind it. So there might be a YouTube video from a previous school where we have discussed all the details. But in short, what you can see in his work is that he writes the free energy for the interaction of the two molecules as a function of a Coulombic contribution and an induced charge contribution. You can see that there's a negative signal here. So the green part is always going to be attractive, always. So even if I have a protein at the isoelectric point, it means that the first term here is going to be 0, but I'm left with an attraction. And the attraction is not short range. You can see that the Coulombic part is long range. It goes to 1 divided by R. And this induced terms is R squared. Of course, if I compare both, the Coulombic part is bigger, is going to be stronger. The green part is going to be weaker, but it's not R6, it's R2, it's R squared. And you can also see here that there is this C's here that has received different names in the literature. Maybe we can say that there is a consensus now that we should call it charge regulation capacity or protein charge regulation, or whatever you prefer to call it. And essentially, it's a fluctuation of the charge as a function of pH. So it's a quantity that you can also get it from a titration plot. So it's a quantity that can be taken from the experiments as well. But look here. If I have one protein that has a net charge equal to 0, one of these guys, let's say B here, is going to be our protein with 0 net charge. So this term is going to disappear. This one is also going to disappear. But again, we are left with this guy here. And depend how big is this C for our B protein, this number can eventually be bigger even than the Coulomb interaction. This number here can be bigger than this one. So this is a way to explain why we can see this attraction that has been received the name on the wrong side of the isoelectric point. So what you saw so far is just a physical equation. But let's see how we can test it in terms of simulations. So what we did initially was to do it for a protein polyelectrolyte system, and we used food proteins. Food proteins were quite nice at this stage because there were lots of experimental data about it, and furthermore, they have more or less the same size, so you're not playing around with your other interactions. I mean, essentially, we can keep more or less the same volume, but they have different physical-chemical properties. So in this case, we test lysozyme, alpha-lac, and beta-lactoglutamine. And doing so, we knew, I mean, we used the model that I showed you yesterday, by the way. And what we did was, okay, let's quantify the free energy as a function of separation distance. And if for these three molecules, we can see attraction when they are at the isoelectric point. So my protein had a net charge equal to 0. I had a polyelectrolyte made of 21 beads. Each bead has a negative charge. So what you observe is like a zero charge protein interacting with a negatively charged polyelectrolyte. And we can see that there is a complexation, because the free energy, as you can see here, is a negative number. So that's what you get in terms of the separation distance. And the order that appears here obeys what you can predict from the Kierkegaard-Schumacher equation. Because when we try to quantify the numbers, I mean, if I take the PI, the isoelectric point, and I take what's the number that I have for the charge regulation capacity, what I have for the dipole interactions. I mean, remember, the net charge is 0, but the dipole is not 0. And if I go from lysozyme, alpha-lac, to beta-lac, we can see that there is an increase in Cs in this direction. The dipole of these two molecules, I mean, you can also see that there's an increase. So in the case of alpha-lac, you expect that ion dipole interactions can also play a role, but the order follows what you can predict if you just look into this contribution here, because all the terms should be 0. One protein has a net charge of 0, so it's gone. Well, the simulations, I mean, they confirm that we can really have something. I mean, of course, we got the PDB structures that was not available for Kierkegaard, and we try in a computer, and just using electrostatic interactions, the model, I mean, the model has nothing except charges and the pH that allow them to fluctuate. Well, remember that I also showed you that one of the hypotheses was that we could have this patch interaction, and there was this idea that these amino acids were responsible for attraction of the oppositely charged amino acids. So we said, well, what about if we make mutations? Because then we can prove it is either important or not. We can do single mutations. We can mutate all these 5 amino acids that were supposed to be responsible. And you can see that even if you do 5 mutations, we can still find an attraction. I mean, it's always negative with this number. So it was a proof that it was not needed to have this amino acid to have a complexation in this case. Well, in computer, you can play God. So a protein with 0 net charge can be written in different ways. One simple way is to say, okay, all my amino acids now have 0 net charge. So 0, 0, 0, 0 plus 0 is going to give you a 0 net charge. We can also go to a protein that has a titration. I can freeze the states and just look to the dipole moment and use the dipole moment to make this protein a 0 net charge protein. We can just keep the fixed charge, so I can include all the high order moments as well. So now we don't have only the dipole interactions, but all the high order terms. And we can have the one with a charge fluctuation, where this pH effect is going to be present. So for the last guy here, we are doing a constant pH simulation. For the others, we are just playing with vanilla simulations, like the charge you fix at the beginning, and they're going to remain constant with no change in their protonation states during the run. Doing so, we can compare, like for lysozyme, a molecule that has a small dipole moment, that has a small number for Cs, and you can see that the only way to observe a complexation is when charge fluctuation is present. So only when I have a constant pH simulation that I'm able to observe the complexation. I mean, this can be seen in different examples, and when you are running calculations that are not constant pH simulations, sometimes you're not going to get the proper behavior of the system because you need this phenomena to have the complexation. So that's another take-home message from these results. On the other hand, I mean, if you look at alpha-lac, in the case of alpha-lac, the molecule has a large dipole moment compared to lysozyme. So in this case, when you compare here the results, if you have just a dipole interactions or the one that comes with charge fluctuations, you can see that the blue and the green line, they are on the top of each other. And actually, this was a reason why in previous works, people could see a complexation for alpha-lac, because alpha-lac is the case where the dipole interactions are really important. So what has been published before was okay, because it happens that for this molecule, the dipole interaction is really quite important. I mean, it's equivalent in some way. So now we know a little bit more about this system, and we know that depending on the profile, I mean, the physical chemical parameters of the net charge, the dipole moment, and disease, we can more or less predict what's going to happen with the system, what interaction is going to be the most relevant one. And in fact, I mean, another thing that we have done, I mean, was to discuss if these interactions were specific or not. So we could use the same trick that I showed you yesterday about the pKa shifts in the presence of another charged molecule. So I have the protein here, the polyelectrolyte is coming around. When the polymer is coming closer to a particular amino acid, it has a charge, and this charge is going to shift the titration properties of this amino acid. And doing so, we can see which place of the surface of the protein that this polyelectrolyte has been visiting. And when you look into where we have the shifts, we can see that they are everywhere. So this is, again, an indication that we have a non-specific interaction. Okay? We have tested many proteins, and one of the intentions to do so was to make sure, I mean, when we could have one interaction larger than the other. So for this set of proteins in particular here, you can see the number of amino acids, you can see the isoelectric constant, the pi, the cis, and the dipole moments. We can see what you get from the simulation results. And using these numbers here and plugging the equation that I just showed you from Tanford, from Kirkwood and Schumacher, you can see how big is each contribution. And looking to this table, you can see that there are cases where most of the time the charge regulation contribution is way larger than the others. In particular, if you look here for lysozyme, the contribution that you have for the charge regulation is 30 times larger than you can get from the ion dipolar interactions. Except for, I should say here, except for albumin, the top one here, is where we have something larger for the ion dipole interactions. So this is a way to show, I mean, what is really the driving force for each of these proteins under the studied conditions. That in this case is at the isoelectric point. Okay, we have this equation, and we can see, I mean, again, the same trick. I mean, we can make the isoelectric points just to get rid of a few terms, and doing so, it becomes easier to identify what's going on. So we can, for instance, play with the charge of the polymer. So instead of having 21 beads, we can increase the size of this polyelectrolyte in such a way that we can see what happens if I increase C, to see if they are also obeying the Kirkwood-Schumacher equation. And it turns out to be really the case. I mean, you can see that we have decreased and increased the size of your polymer. So now we have here, in this last guy here, 51 beads with -1. And we are moving in this direction here. So from here to there, we can see that there's an increase in attraction. What we have in this plot is not the free energy, because now we are not trying to make more fancy simulations. I forgot to mention, but when we are trying to get free energy, even for a simple system like that, you have to use metadynamics, because free energy is really very difficult or very painful, very costly, whatever you prefer to use to get the numbers. So in our case, we are not really trying to get a number that can be quantitatively compared with experimental data, but just a description of the attraction, if you have or not attraction. So it turns out that you can simplify a lot, and also to simplify the simulation, if instead of looking to the free energy, you look here to the radial distribution functions, and you multiply this by the square of the separation distance. So in this case, when you have a plot like this one, you are in the conditions where the monomers try to be far away from the protein. And as much as you decrease this peak and move it to the left direction, you are under the condition that they like to come very close to the protein. So we can see this change in the conformation, I mean, they are the configurations, and that in this case, they are not happy to be close, and here they are happy to be close to each other. So this is where we have a complexation, and here they are just apart to each other. The two molecules are apart to each other. Actually, this is a very nice trick to know, because this simplifies a lot. So if you're just trying to describe the phenomena, it's a very good trick. Of course, it's not going to provide you a quantity that can be directly be compared to experiments, except the behavior. We have tried, different things, even also the pH effect. So this is also some snapshots from the simulation that they also tell us a little bit more what's going on. Because although we have attraction in these charge regulation conditions, you can see that's very different from what you have when really the molecules, they are in the full, let's call full attraction regime. Because here, they have opposite charge. So the polymer is really wrapping around the protein in a much more compact way. And here we have part of the polymer that's very close to the protein, but some parts of the polyelectrolyte might be just floating around. So this is also another difference between the two conditions. And in the case of repulsion, of course, they are apart from each other. So you can see the transition between the same plot, like is the rate distribution functions times to the R square. Here is the condition that there is no complexation, and here we go to the direction where there is a complexation. As high as this peak, stronger is going to be the complexation. Especially for the case where we have been studying this and calculating the free energies, we can really make this directly comparison, because these are exactly the same system that I showed you in the first slide, where we have the free energy as a function of separation distance. So that's why we can really demonstrate that this quantity can be useful with a lower CPU cost. So just to show again the regimes. Another thing that we can do, you can look at this from the perspective of the polymer. We have been looking to different quantities. What kind of measurements can be good to quantify these interactions? Let's say, in a way that they are less costly, in particular if you like to explore, again, this on a large scale. You're trying different mutations, different salt conditions, different pH conditions. And it turns out that you can look into the polymer. A single polymer isolated in the solution is going to give you this dashed line. So this is a reference. So a polymer that's not interacting with a protein would behave with a radius of duration as the one that you can see in the dashed gray line. Looking to what happened in different systems, you can see when the molecules are close to each other, so they behave like his. They have a smaller radius of duration. And they should have it because they are really very close and they are deformed by the attraction that they have with the protein. As much as they are leaving, we are going to conditions, pH conditions where they get far away, you start to see that the number is going to approach, going from here to here is going to approach what you have when they are isolated, which makes sense. If the polymer is far away from the protein, it has a behavior equivalent to be just isolated in the solution. So that's another perspective. You can look both to the rates of durations or to end-to-end separation distance. Well, there are many, we have done analysis of a couple of systems, at least several of them, I would say, I think about 10 different systems. And here's another example about protein-RNA interactions. So in this case, you can see that this protein and the RNA molecule is negatively, they are both negatively charged. So if you don't have charge regulation, there's going to be a repulsion, free energy as a function of separation distance. But when we switch on charge fluctuation, you start to see an attraction. So it's well known that these molecules are going to form a complex, and it was something that was hard to explain if your RNA molecule and your protein, they have a charge with the same signal. And we can see that there is a change in the behavior of the free energy if you switch on the charge fluctuation. So it could be that charge fluctuation is not enough to give an attraction. There might be other things happening in the system, but at least it's part of the solution and it should be there and it's going to give you at least some attraction, as you can see here. Okay, I think I have, I said most of the things I'd like to say about this problem, but I should also mention that there are, of course, other labs working with similar ideas. This is one paper from the guys from in the Netherlands, where they use immune field theory and they show more or less the same thing. But there was a problem. Very often we have these two views. One is the charge regulation and the other is the PET theory. So both, let's say, hypotheses have been around for, I would say, about 20 years. And it turns out that we could finally a good way to merge these two things. So in this paper, we demonstrated that if you look into the properties, how your parameters are distributed for your given molecule, one or the other interactions can be the most relevant. I mean, without going into all the details, what is important is, like, first of all, you fully characterize your protein or your polyelectrolyte. So when you get all these charges as a function of pH, all these C's, all these dipolar moments, and if you have this information, you can analyze and see if they are case 1 or 2 or case 3 and 4, and knowing this information, you can see if the driving force will be mainly charge fluctuation or to be mainly the ion-dipole interactions. So there is a way to get rid of this problem if you first just do this initial analysis. Now, I think it's good that we change a little bit the gears and go for protein-protein interactions and make these more applied things. There are a couple of examples that I mentioned in my first class, so I'm going back to them and tell you a little bit more about these things. One model that we're going to be using a lot here is what we call this 40, is where we have 2 proteins. These 2 proteins are free to translate. They are free to rotate in any directions. And as you can see here, pH is part of the game. So it's coupled with our FPTS. So that's the way that we control pH. Essentially, we have this function where pH is an input parameter. We have the pKa's. Just to go back to my second class. And doing so, we are going to sample the system, and sample the system, we are going to get the free energies. So that's one way to calculate the free energy. And by the way, since we have a coarse-grained model, we smooth the landscape, the energetic landscape, which means that it starts to be doable to extract the free energy of the system without playing all the tricks. So that's something that can be nice. I mean, of course, the results are not going to be numerically comparable with the real data, but at least you can get things that are the same order of magnitude, or you can use to extract relative binding affinity. That's the case that more often we are after. And with another extra advantage, because as you can see, I can do this at different pH and salt conditions. So it's something nice. We have applied to several systems. So back to what I was describing to you at the very beginning, this can be used in different systems, but we decided to apply more or less 15 years ago to virus. Our initial motivation to study virus was not, in fact, the virus itself, but it was the fact that there was an idea that if you compare an interface, in general, protein-protein interface, and the interface where we have a virus, they have more titrable groups. So especially if you look to an antibody and epitopes interactions, you can see that they have more tyrosines. You probably remember that Elias showed you this, more or less the same data yesterday, or if not, you can talk with him, and you can see, for instance, there is a high tendency to have tyrosines. So if you have titrable groups, it's already an indication that electrostatics should be important for these systems. And I'm going to show you very nice results how electrostatics seems to play a role in these systems. First of all, we are not the only ones to say such things. I mean, there were papers like this one that have been published for flavivirus, and you can see that they even use electrostatic maps to make a difference between dengue 1 and dengue 2, West Nile virus and Zika virus. Just look at the profiles. This was an experimental paper where they demonstrated it. You know, we already got inspired by this paper to demonstrate, for instance, the difference between the strains that we have in Brazil for Zika and the ones that we have in Uganda. We have analyzed different ones here, and you can see that, in fact, they do have lots of difference in the electrostatic features. So this is somehow connected how your immune system is going to respond to this virus, because now you have another protein with another net charge. But I'm not going to talk about flavivirus. This is what I did, I guess, in the previous school. So we changed gears to SARS-CoV-2. That actually was one of the examples that I gave in March 2020 here. We were in the very beginning of this work. It was the beginning of the pandemic, and we were starting to work with it. At that moment, I mean, January 2020, this was the information that we had. I mean, we had this communication from the World Health Organization that something different, I mean, a different phenomena was appearing in China, and there was unknown cause. 65 days later, if I'm not wrong, we got the full information about the proteins, I mean, the sequence of the proteins that were involved in this, let's call, new virus. And we could see that there was a spike protein, and it was pretty much close to what we knew from what was called at that time SARS-CoV-1. Sorry, just simple SARS-CoV. So we took this paper, and we asked ourselves, okay, does SARS-CoV-2 use the same entry mechanism that we know for the old SARS-CoV? So what was the entering door of this new bug? Was the same exactly what we had before? Oops, sorry, something wrong happened here. And if so, how the ACE2 receptor that was used for SARS-CoV-1 would compare in terms of binding affinity? I mean, if you compare the two bugs, what would be the difference? If so, can we think about a way to block it? What about to design an antibody? Or what about antibodies that were known to bind to SARS-CoV-1? Would they work with this new bug? So this was the motivation that we had in January 2020. The information that was available was this crystal structure from SARS-CoV-1, I mean the receptor bind domain, and the ACE2. That's the PDB that we had at that moment. And we suspected, I mean, this was not even published at that time, but we suspected it would be similar, that the spike protein would have the receptor bind domain, and this could be really the entering door. So we made the hypothesis that, okay, SARS-CoV-2 works as SARS-CoV-1, so let's try to see if the receptor bind domain are going to have a strong attraction for ACE2. This was also something new at that time, because very often you probably have this feeling when you could hear people that were doing theoretical studies. Often we have experimental data, and we do simulation, then we compare with the experimental data, and if there's an agreement, we are happy, and then eventually make a prediction. At this time, we didn't have any experimental data. So we have inverted the order. I mean, we needed to publish something to predict, but we could not validate. So the experiments, they came later. Actually, it was a chaos, because at the same time, everybody was rushing. So during the review process of the paper, you can see that we have been changing and introducing. So if you go to the bioarchive, for instance, where we published the first version of the paper, there were no comparison of experimental data. And then in the final paper, we finally have a comparison. Because the thing is, we didn't have enough information. I mean, it was just a hypothesis like to try. So it was a moment to challenge the simulation methods. So it was something bad that was happening with us, but at the same time, it was an opportunity to challenge the models. And the referees were not unhappy because you were not comparing with experimental data. Simply, there were no experimental data to compare, at least at the very beginning. So we used this as information. By the way, AlphaFold was not available. So now we are back in time that the only way to try to prepare a structure that we need as input for the simulation was using modular, homological modeling. And in fact, that's what we did here. You can see the superposition of the two structures, the one from SARS-CoV-1 and the one from SARS-CoV-2 that was created using modular. The difference between them has an RMSD of 0.64. Structures that appeared later, I mean, after the publication of this paper, if you compare different structures, they are about the same range. So we know that for different structures, they should be, for the receptor bind domain, should be around 0.6. So we did something that was reasonable, I would say. And using this structure and applying the model that I just showed you, this Forte simulation, where the proteins can be translated and rotated using pH, we could see that the free energies as a function of the separation distance were more or less equivalent. So this was not the first, because there is a paper that later we realized that was published 2 days before us, but it was the first paper where free energy was calculated for such system. There were many, many other papers. We wrote a review paper at the beginning of the year where we make the historical review of all this process. And there was an explosion of a paper just after this one. But this was, using binding affinity, the first paper that could demonstrate that the entering door of SARS-CoV-2 was in fact ACE2. So this was nice. The other thing that we could calculate is the number of amino acids that were involved, and we can compare between the two viruses. And we can see that for SARS-CoV-2, there were more amino acids that were important for the interaction of AC2. The way that we are calculating is using Proceed pKa that I introduced it to you yesterday, and is essentially that analysis of the shifts in pKa's when you have a molecule coming around. So, okay, that's nice. How does this compare to experiments? Well, later there were lots of experimental data. For this analysis here, there was this paper. I mean, the numbers, of course, is not exactly the same, but the same trend can be observed in the experiments that were published after we have made this analysis. So again, this was very nice because we could really validate the method after we have done the work, not what is, let's say, the traditional procedure. In terms of the binding affinity, I mean, you can see that the numbers, they can be quite scattered, so it's harder to know if it is right or wrong, but at least they are equivalent, we can say, in a more, let's say, safer way. One reason why you have all this diversity in the results is because often the physical chemical conditions that are used in the experiments are not exactly the same. So if you change the salt concentration, if you work with a dimer of AC2 and not a monomer, if you have all these changes, it's harder to make really a directly comparison because the conditions are not exactly the same. But anyway, they give you an idea about the feeling. So we could use it. That's the result that I already showed you at the very beginning, but I didn't make some comments about it. I made the comment about the linearization, but there is something else. What's nice is also that we can see that when you do this analysis, what you extract from your analysis is the Develio theory. So what we are getting here is that if you look at the linear regression here and you take the coefficients of this line, this number is what you are going to plug here, and that's the number that if you just take the charge, you are going to take to get anyway. So you can recover a physical theory just using the computational data. Moreover, this is for me one of the beautiful results in my career is to see how nature is exploring electrostatic interactions. So what we have here is a correlation network. Essentially, we have calculated pKa's for all the variants. We use the white type as a reference, and we calculate the difference. And we try to see how they cluster to each other. When you do this analysis for the variants that were appearing over the time, you can see that we have, for instance, look here, we have gamma, we have beta, we have the Omicron variants, and I cannot see from here, but there should be a block with delta, it's here. So you can see that the variants, they cluster. And the way to do this analysis is simply using pKa's, the same pKa's that I discussed with you in the first class. If you compare this with a phylogenetic tree, we get a phylogenetic tree. Just look again, I mean, you have the gamma, the alpha beta like in green, we have the deltas over there, and you have the Omicron variants. So using pKa's, using a physical chemical quantity that's directly related to electrostatic interactions, you can recover the phylogenetic tree of the virus. So this is, for me, is amazing because it shows how biological science and physics, they can really talk to each other and they go together. Well, the second question that we asked at that time was about how to block the virus. I mean, we knew that there were 4 antibodies that could bind to SARS-CoV-1. These are the 4 of them. So we said, what about if you try them and see if they have some chance to neutralize this virus? Just remember also that neutralization is one thing, binding is another. First, you can, I mean, in the computers, we can assess the binding, but we cannot really assess if the binding is going to be enough to neutralize the virus. That's another story. But at least if they have a potential to bind to the virus, it can suggest that eventually they can be used for neutralization. So we tried the 4 of them. That time there were just 4. If you go to the protein data bank nowadays, the number of cases that you can find of antibodies in undercomplex with different RBDs from different variants is a very large number. I mean, I don't remember even now. I think it was more than 1,000 the last time that I looked for it. So we did the same thing, and then we compared the 4 cases, and it turns out that this CR3022 could be the best candidate. You can see that there was a slight difference. I mean, the same antibody would bind with more attraction to SARS-CoV-1, but at least it was the one that could come in close. So out of these 4, CR3022 could be a good candidate. So that's why we started working with this guy later. Again, We did the calculation. There was no experimental data to compare. Later, we were very happy to see that was the case. I mean, there was this paper that showed that this could be possible, the binding with this guy. And there was also a structural result. So again, they confirmed that the same antibody could bind. You can see the crystal data here. Okay, very nice. So let's see. Okay, it's binding, but we'd like to improve the binding. So now I'm back to protein engineering. We created the initial different steps how we can try to optimize this guy. So essentially, we are taking and using our tools to try, I mean, all these quantities, I mean, affinity, to predict the epitopes, etc., blah, 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 aggregation, stability, and making mutations, making alanine scan. We can go one by one and test if that particular mutation could improve these quantities or not. So, for instance, if you take this sequence here, we could replace it by different combinations where you can see that I'm just changing one by one the amino acids and trying to observe what happened. I mean, which case that the binding was favorable and which case the binding was not going to be in the right direction. We have done this loop process in this pipeline for 3 times. So we could check up to 3 mutations, the ones that could improve the binding affinity. And doing so, we proposed some changes in the CR3022, that's the priming one, the mutant one. And here you can see the amino acids, the titrable ones that would like to neutralize or to change in such a way that could improve the binding affinity. And with these 3 mutations, you can see that we are able to go from here to more or less an equivalent binding affinity with the old virus. So it was promising. Come on, now we know the entering door, which can help people to design vaccines, because now they know that it could be really nice to target the virus. And the number of evidence for this starts to accumulate. That's why most of the vaccines were targeting the spike protein. We also know that there are candidates that can bind to the virus and eventually can block it, and we can even try to improve it. Of course, we are happy with it, but we are not totally happy. I mean, we would like to do something even better. So we try a multi-approach. So we combine the different strategies. First of all, we started here with Rosetta. So we started looking to the CR3022, and we asked ourselves if we can find anything better than this interface. So we know the crystal data at this point. I mean, the crystal data was known. And now knowing the crystal data, what about if I can find a solution that's going to give me a better contact between the 2 molecules with an increased binding affinity? So we have done analysis with Rosetta. We produce a large number of poses, I mean, or docking results, if you prefer to say so. And you know that the scoring function is not really ideal in this. You probably remember what Elias showed you yesterday. So we were aware of that problem and would like to get something out. But we say, okay, what about if you try to have a consensus? And let's try to get just the top candidates and make a better analysis. So we started with Rosetta. We took the top 10 candidates from Rosetta, from the Rosetta units, energetic units, and we defined this criterion. Let's only select the guys that they have energy that's more favorable than what we know is going to be the case for the original molecule, for the original complex. And also, we'd like the interface to be larger. So we created this criterion in terms of the surface accessibility and also the energy of the system. And we also look visually to avoid, I mean, if there was any kind of overlap or steric problem with the structures. And using these 10 candidates, we decided to make a very, or let's say a more complete analysis involving different things. One of the things was running a constant, not a constant charge simulation, not constant pH. So here we fix the charge and use a coarse-grained molecular dynamics simulation with CETA force field to get the free energy using metadynamics, very expensive simulations. We also have done some analysis to check the results using our Altum simulations. And we have used our own approach and tried to see if we could find a consensus. So with this pipeline here, and also introducing here the semi-electrostatic optimization that I just showed you. So in this combination of things, we could see how could be the best candidate. And we could also test this guy, not just now against the wild-type version of the virus, but all the set of variants that happen to appear on the way. Because over the years, I mean, every half a year, more or less, a new variant was taking care of the place. So here, we could go from the case where we know the results, from the case that we start to explore and see if what we are proposing as a new candidate could really work for the RBDs that were available. One nice result was that, okay, we could find a consensus. You can see the results from the 3 approaches. The Rosetta, you can see the consensus here. You can see the Rosetta data in green. You can see the CETA force field, the constant charge simulations in red, and our approach, the constant pH simulation in black. Remember that FORT is the most coarse grain approach in this case. So it's where you have all, let's say, simplifications of the model. And we are lucky to observe that for P1, that's the candidate that we observed, and this guy is the best one according to all the methods. P6 was equivalent in terms of the FORT simulation, because in this case, we can probably have a lower statistical errors. We run longer the simulation, so we decrease this guy. You can see that the error bars for the constant charge simulation with CETA is much larger than what we get in the simulations, because you cannot afford to run that longer simulations. But another good result here is, first, we could improve. You can see that the wild type is here. And now we have something that has a higher binding affinity. So this was also something very positive. And another thing that was nice, in particular for us that have been working a lot with 40, is to see that you get, in a cheaper way, the same thing that you can get for more sophisticated analysis. So that was also cool. We tested these antibodies with different variants, and that was also something even more surprising, because they work with all the other variants. So we started here with the previous antibody. That's the result that you get from Rosetta. And now we try to go further, and we can see that they are also going to work even with BA.21. That's the omicron that you can see here. And they also have a higher binding affinity. So this was cool. Another thing that we have observed is that, well, what about the driving force? And you can see in this top plot here that the charges of these molecules, more or less, they have some relationship with the binding affinity. So again, we observed that the charges were very important for the receptor binding domain to interact with the cell, but they also seem to be very important for the interaction with the antibody. So this is why we're all the time trying to explore electrostatic interactions for the antibody designing or engineering, because it seems that it's hidden there, the information. Okay, we were happy, but there is still work to do. We have colleagues that are trying now to do the experiments to really assess if these molecules are going to work. We had some preliminary data that were not so good, but now we are working a little bit more to see if we can get more data. In particular, we are trying to get cryo-EM data of these molecules to make sure that they are binding on the right position and this kind of thing. So eventually, this can be further tested and further validated. In parallel, I'm just going to mention very briefly this. We are also running a project that now we are trying to design the same things, but using machine learning. This is a collaboration with the lab from Animesh Ray in the US, and they are doing the analysis using machine learning, and we are testing it using the simulations, particularly with 40. And we were very happy and even more excited, because now we got a couple of sequences that when we test the binding affinities, they are better than what we could design only with simulations. So at the very beginning, I was showing you the troubles that we have with the machine learning approach when I was describing you what the troubles with AlphaFold. I mean, or points that you have to keep in mind when you are using AlphaFold results. Now I'm showing the opposite. I'm showing that machine learning, in some cases at least, can offer you something better than what you can get, even if it is very complex or combination of methods like the pipeline that I was showing you. So in this case, you can see that the molecules that are coming here, they have higher affinity than what we get before. I mean, these are the dark blue ones are the ones from the machine learning, and the ones in the light blue or green, whatever you can see from here, it's the one from just the simulation part. There is even something even better with the machine learning approach, because we know that these molecules are available. I mean, they are molecules that exist in real life. And in our case, we don't know if these molecules are going to survive in real life. What about their stability? I mean, what about when you try to purify them? Because if you use the DNA to make the purification process, later, eventually the conditions can be so harsh that eventually we're going to destroy your molecule. So in this case, we know that they exist, and we know that they have a better binding affinity. So this is something very promising. Also, when we check the stability using electrostatic interactions, you can see that they're, in general, more stable. I mean, there are cases that are not so stable, but in general, they are more stable, which is also promising. Before I finish, there is something very important, because we have been talking about protein engineer, protein design, and very often my students ask me something. Hey, what about results, practical results? Because very often we can hear papers that, oh, docking studies, a new molecule, blah, blah, blah. So there are hundreds of papers that you are promising to solve the problem, and at the end of the day, I don't see these products in the markets. So you have shown something very nice, promising, but are they going to work in real life, in the wet lab? Do they really have a potential for something? I described to you that we are going to test them. But very luckily, they are not going to not work in the first moment, because we have to keep in mind that there is this cycle here of the process. You design something, you test it, and then you redesign. So this is a loopy process. Back to the question about the docking for small organic molecules, there are around 8-9 cases where it has been seen that the computers were able to design something. But we have to remember that most of the time what we get out from the simulation studies is knowledge. Knowledge that can be used to speed up the process. I don't know about any single experiment that you can do in the wet lab that can provide you with a new drug. Very often we combine the results, we combine knowledge to help us to move it further. And this is also the case when we're designing the computer. So try to remember about this, because otherwise you can be a bit frustrated. I mean, if you get a result that was promising, your result should give you the drug of the year, you're going to get a Nobel Prize. And in fact, I mean, this is a loopy process, and that if you succeed many times, of course, celebrate. But you cannot celebrate for each individual step, because you're always providing knowledge. This is something to keep in mind, and especially you are the future of this field, and it's important to remember that you need to be patient with this process. We are providing knowledge, and that's all at this moment. Luckily, the molecules can be elusive. If it happens, next school maybe I can tell you how was the story in a different perspective, but so far this is the point. And also there is something that, at least my opinion, I mean, just to try to conclude the whole thing, I mean, I try to show to you that machine learning can be powerful. That's the last example. But at the same time, I try to warn you about the use. And there is something that I don't know if it is clear to me, that we still need data. And there are two ways to get the data: experiments and simulations. So for a while, I mean, of course, it's going to be nice to see the explosion of all these machine things, and there's a long future. But in my personal opinion, and I don't know if my colleagues are going to share with me, but at some point, if you're not able to produce more data, we are going to reach a plateau. And if you don't have more information, we are done. So we need to keep in mind that we still need to produce data, experimental data, and theoretical data, so we can feed up the machine learning work and be able to do something useful. So that's my, at least opinion, my warning to you to don't forget that the traditional tools are still valid and is still important in this context. I think this is a good way to close. So this was not in my remarks here, but it's something that I think is important for a pedagogical point of view. Just to summarize, I mean, I try to summarize all the details that I have been sharing with you, but I hope that I could demonstrate that simplified model can play a role even to design new molecule or to help you to rationalize process. That electrostatic interactions can be really important. I gave you a couple of examples. Charge fluctuation is a mechanism that, when it's absent, is not going to give you the conditions to reproduce the experimental data, and it's often neglecting the simulations. So very often, when people are running simulations, let's say, the best that they do is to start with a molecule, they run PropKA, H++, whatever tools they use, and they get the charges for a given pH, and then they forget about protonation state. Charges are going to remain constant during all the run, and you hope that this is going to be true. It can be fine for most of the case, maybe, or at least some of the case, but there are situations that is not going to work. Lysozyme is a good example of it. And I also hope that I can demonstrate that we can use this kind of ideas for antibody engineer and eventually any kind of interface. Data that I showed you came from different places and different collaborations. I mean, the list of names, as you can see here, is quite large. The founding people is also quite large. Number of students not that big, so that's something if you'd like to join us to Fort, you are also welcome. Maybe your name can be here in the next slide. But we can never forget to say thanks also to the people, to the taxpayers, because without their taxes, we don't have this money and we cannot have the research. So this is something also important to remember. And that's, I think, is the end of my presentations. I thank you for your time. I thank you for coming here on a Saturday morning before a tough week of lots of activities. And I'm really happy to discuss anything with you and answer questions. So thank you. Yes? What do you mean by generic study? Well, what we have done, I mean, is initially was to study how charge and dipole moments were evolved for the different variants. So we could see that over the time, it was increasing the number of positive charge and the net charge. So this is something that we have seen before. And then at some point, I said, oh, come on, what about the distribution of the charge in this protein? And that's how we came out of the pKa analysis and these things. So this is all the analysis. We can also see that for the receptor bind domain, something that I mentioned, I think yesterday for sure, but I mean, I don't know if the first or the second class, but what I mentioned was the coupling, the electrostatic coupling between the amino acids. And another thing, I mean, in this direction, what we observe is that the coupling is quite large as we are evolving in time. So for the new variants, it seems that more charges can talk to each other.
So you can actually see the regions of the protein that are undergoing the fresh selection.
Yes. Yeah, so this is nice. And I mean, there are data that I'm not showing here, but we are able to predict, I mean, how far we can go now. And it's, unfortunately, it seems that the virus can still explore more, at least for the entering process. There might be other, let's say, parts of the viral cycle is going to prevent us from more trouble, but there is still room for the virus to evolve and be more transmissible. Because another thing that can be shown is that the relationship between this attraction of the ACE2 correlates or has a relationship, maybe it's better to say it this way, with the transmission. So as we are moving from the wild type to alpha, beta, and so on to the Omicron family, we can see that the transmission has been increasing. If you calculate this R0 coefficients. Yeah. Yeah, it's, I mean, nature is able to explore what's available. And we can also see that there is a tendency now for the last variants that they also start playing with the dipole interactions. So far, it's just charge interactions. But the new variants, they also start looking, I mean, you can see that there is a slight increase in the dipole moments. So maybe we have reached a plateau where charge is not enough. And also, after all these mutations, we have to remember that the protein has to remain stable. And it's actually also another surprise, I mean, because we can see that even sometimes a large number, like 20 mutations, they are out of 200, more or less, that's what you have in the receptor binding domain, it's not enough to destroy the protein. So we are afraid to do more than 3 mutations for the antibody molecule. And RBD can take 20 with no troubles to keep the structure. It's amazing. I mean, it's amazing from, let's say, from a scientific point of view, but it's a trouble if you were the victim, right? And I can tell from my own experience that's not nice. Yeah. But it was really very nice, I mean, that we can see, I mean, where are the changes? And how is the coupling, which also helps to understand what I introduced very briefly. I didn't discuss this in deep details with you, but often we have this lock and key mechanism. So when we define epitopes, we are all the time concentrate at the contact. And we have named electrostatic epitopes because we can see that it goes much more inside the protein structure than reached at the interface. So a mutation that's more on the other side sometimes is going to affect a lot the interface. And if you just focus, yeah, because you have all this communication, like it's a kind of chain of events. So you do a change somewhere else, and it's going to propagate to the interface. So sometimes you lose a bit the affinity, and the mutation was not at the interface, but it was far away. But the common sense in biological science is only to look at the interface. What we have seen is like the interface not enough. So that's why we name it electrostatic epitopes, to make sure that we have included the ones that really participate in the game. Yeah. Was it that you were asking for? Okay, thank you. Thank you. Any more question? Yes, please. Schumacher. Well, that's the Brazilian pronunciation. I don't know if in German it would be the same. Yeah. You mean the ionic strength? Yes. I haven't tried, but I guess not. I mean, maybe they can give some small numbers, but I'm not sure, and I haven't done any analysis like that. I mean, we're happy to look to the level, and we are happy because, as you saw, the simulations were conforming the attraction, and that was, I mean, essentially what we were using. I mean, we used the term for the Kirk more or less as an inspiration, so it was like, okay, the ideas of charge fluctuation is there, and with this idea, what we can do in terms of simulation. So we didn't really look very deep into all the details that you can extract with the Kirk-Schumacher equation. But I guess it's not going to be a big number. I mean, yeah, I think, yeah. Maybe, I mean, looking to these small corrections, could it be good if you would like to do more analytical results? Because in this case, instead of running more expensive simulations, we could, in principle, just simulate the titration behavior of a single molecule and use this information as input in the Kirk-Schumacher equation and predict how it's going to be everything. In fact, one of the tables that I showed here, where we have all this analysis, if you compare what you're getting, and I can go back to, just to make it clear. I don't know what happened. Maybe not. Okay, never mind. So one of the, there was a table that I had a set of proteins, and for this set of proteins, we calculated the different contributions. If I take the results that we predict from the Kirk-Schumacher equation and the results from the simulations, there is a nice relationship between them. And we got a line with a regression coefficient around 0.9, something like that. So probably we can improve it if we include other higher order terms. That's my feeling. Maybe something that we can try. Yeah, free energy is a quantity that when you try to simulate, in particular if you have a landscape with lots of energetic barriers, it's harder to cross them. So you have to run and run and run and run, or use the tricks to try to pass these barriers. So one trick like that we use in the case of the protein polyelectrolyte is to use what Gunnar Karlström called penalty function. So we start penalize the more often observed configurations, and then we use this in the Hamiltonian to try to decrease the chance for them to appear again. You can use it as well, but there are different strategies. Like we have used in the other metadynamics. That's essentially, there are different strategies, but even so, they're much more costly in terms of CPU. So if you can use like, if I'm not after to get really free energy, but I would like just to see the behavior of the system, using this trick that you use the radial distribution functions times the square of the separation distance, it provides you how is the distribution in a much cheaper way. And that's enough for this discussion, right? Anyway, the results that we are getting from this kind of coarse random model, they're not going to be the same scale that you can make a comparison with experimental data anyway. So often the comparison is qualitative anyway. So if that's the point to make a qualitative comparison, why am I going to spend more resources if you can get something in a cheaper price? Why we pay 3 times the price in a plate at the hotel if you can have it here in the cantina by 1 divided by 3, right? Any more questions, comments? I guess you were all tired, right? There was even a picture about you that I was hoping was not going to happen. Oops. This is weird, but you see it's not working. But there was a lion here that was tired. I hope you were not like that. And I think Rolf and I, we can close the school, right, Rolf? Yeah. No way. I think they are closing us. Yeah, I don't know what's going on. Here it's working. But anyway, never mind. It was just to show a lion. If you want to say something, or should I say? Or both? Oh.
Well, thank you very much all for coming and staying until the last day, Saturday morning, and for the many interesting questions and discussions. I really hope you have something you take home from this week. And yeah, who knows, maybe we see each other again at some future school here. And also, I would like to thank all the lecturers, actually. I think only Oliver is here, so...
To represent all the others.
Exactly, representing all the others.